Hello, are there any questions? Can somebody confirm that you can hear me?
Yep.
All right, great. Share my screen. Oh, still there. All right, you should be able to see my screen now. Today is April 28th, almost the end of April, and we're supposed to be covering chapter 4. Today in the lab, we'll cover lab 6, and I'll talk more about that in the lab. Any questions about what we're doing? All right. If not, let's open up chapter 4. So I talked about damaging prokaryotic cells and mentioned that gram positives were a little more sensitive to damage than gram negatives. And this is particularly true when we're talking about the antibiotic penicillin G. And the reason for that is penicillin G cannot get through the outer membrane of the gram negative cell. There are other antibiotics that can get through the outer membrane. And as I mentioned, ampicillin, one of the early derivatives of the penicillin family, is a member of the penicillin family, which can get through the outer membrane of a gram-negative cell. There are other antibiotics that can get through the outer membrane as well. And streptomycin is one. And it can get through the outer membrane because it's a lipid-soluble antibiotic. So it can go right through the outer membrane. All right, let's talk a little bit about the plasma membrane. It's what separates the cell from what's inside and what's outside the cell. And it is a little bit confusing if you think about it. We consider the cell wall to be a part of the cell, but yet it is outside of the cell membrane. So just remember that the cell membrane separates the inside and the outside of the cell, with the exception of the cell wall, because we do consider the cell wall to be a part of the cell. The plasma membrane is inside the cell wall. It's mainly made up of the molecule of phospholipid, and we have two layers of phospholipid molecules, which make up the lipid bilayer of the plasma membrane. And what gives the membrane the ability to select what molecules can come in and what can come out is the fatty acid portion of this bilayer. Anything that does not dissolve in lipids cannot move through the cell membrane without a protein helping it. For example, the ions of sodium and chloride are pretty small. And even the hydrogen ion is really small. It's the smallest ion. And it cannot get through the lipid bilayer without coming through a protein. Now, in the plasma membrane, we have a lot of proteins. Some of them are transport proteins or channel proteins. And the difference between a channel protein, I guess, and a transport protein is a channel protein puts a pore in the membrane, and then things can move through that channel. And usually the channel protein isn't specific, meaning it can allow many different molecules to move through it. Whereas the transport protein is very specific, and it allows generally only one molecule to enter or exit the membrane. Now, these proteins can be broken into three groups that are in the plasma membrane. You can call them peripheral proteins, and that'll be a protein that's mainly on one side or the other of the plasma membrane. There are also integral proteins and transmembrane proteins, and both of these proteins span the membrane. All transport proteins will be a a protein that spans the membrane. And yeah.
Dr. Volpe, so in turn, integral proteins and transmembrane proteins, are those the same thing?
Not exactly. And you don't need to know the specific differences, because to tell you the truth, I'm not sure what those differences are, but both of them span the membrane. And that's the only thing we're going to talk about. And the peripheral protein is mostly on one side of the membrane or the other side.
So are those embedded in the membrane, integral protein and transmembrane?
Transmembranes are totally embedded in the membrane. And besides that, they span the membrane, meaning there's a portion of the protein on one side and a portion of the protein on the other side.
Okay, thank you.
Okay, that's what I mean by span the membrane. Whereas the transmembrane, the peripheral proteins are mainly either outside or inside the membrane, but they can have a portion of that protein in the membrane. Okay, does that make sense? All right, hopefully that makes sense. All right. And remember that the phospholipid molecule is the main molecule in the cell membrane, or called the plasma membrane. And the phosphate heads, which are polar, face inside the cell and outside the cell. And the polar heads are facing the water. Think of this being a red blood cell. It's mostly water outside, and the polar head is facing the water. And then it's mostly cytoplasm, which is water inside the cell. And so the polar head is facing the water. And then the fatty acid portion of the proteins, phospholipids, are inside the membrane. Any question about any of that? All right, this is something that always amazes me, and that is the cell membrane is actually a fluid. And we have the fluid mosaic model to discuss how the membrane works. The membrane is a mosaic of phospholipids and proteins, and these phospholipids and most of the proteins can move around in the membrane because it is as fluid as olive oil. If you don't know, take olive oil at room temperature, and it's all liquid. In fact, you put olive oil at the body temperature, which is closer to 37 degrees Celsius or 98.6, and it's even more runny. The proteins move about to function. And those of you who've taken A and P, you know that when a receptor protein binds to a membrane on the surface of a cell, that those membranes move together. Not the membranes, the receptor bound proteins move together. And then that portion of the membrane will be brought into the cell. The point is the proteins can move to do their function. And they are not, we should say many of them, if not most of them, are free to move about in the membrane. The only exception are the proteins which are anchored in place. And a protein which is bound to the extracellular matrix or to the intracellular matrix, and we'll talk a little bit about the intracellular matrix later. It's a network of proteins running throughout the cell. If it's bound to one of the matrices, that protein will not move. So think of this as being, I don't know, like a protein, a cell in the bone. And we have a protein here which binds to the matrix of the bone. This protein is not going to move in the membrane because it's held in place by the extracellular matrix. And the same thing with the protein. We'll take, I don't know, I don't have another good one, so we'll take this one again. If this one is bound to the intracellular matrix of proteins inside the cell, this protein is not going to move about in the membrane. But all of the other proteins, like the transport proteins and the receptor proteins, are free to move about in the membrane. And what's amazing about it is when you look at your hand, it's mainly held together by the cell membrane, because that's all animals have. And it's amazing to think that your hand is being held together by a fluid. That's the cell membrane. Any question about any of that? All right. And then the phospholipids do rotate around so they can spin this way, but they can literally move about this way. And that is actually how water, if it's not getting through through a protein, how water enters a cell. And let's say it's entering the cell, so we'll say it's this way. What the water molecule does is it'll be outside the cell, and it just waits for the phospholipid molecule to move over. And then the water sneaks in that hole before another phospholipid fills up the hole. And then the water just waits right here because it can't get through this side of the membrane. And what it's doing is it's waiting for one of the phospholipids here to move, and then the water can get into the cell. Okay, and that's not a hole in the membrane, because when the water moves this way, the phospholipid moves over, and then that seals the membrane. Any question about any of that? In root cells, plants have proteins that bring the water directly into the cell. And that's to bring in water quickly, because water doesn't move through the cell membrane very quickly when the water outside here is just waiting for a phospholipid to move over, and then it sneaks in, and then it's held in the, I don't know, the first layer or the bilayer, and then it waits for an opening when the second phospholipid in the second bilayer moves over. That would be a very slow way for water to enter the cell. And then the roots, they have proteins to bring in the water directly. All right, the plasma membrane shows selective permeability. It allows the passage of some molecules, but not others. No large molecules can move across the plasma membrane without a transport protein. Ions and small molecules like water either do not move across the membrane, or they move very slowly. Like water moves very slowly across the cell membrane. Lipid molecules can get through the lipid fatty acid bilayer, so they can move across the membrane. But if you think about it, like think about a red blood cell, there aren't going to be very many lipids floating around in your blood because your blood is mostly water, right? But there are some lipids, like there's some cholesterol in your blood, and there's, what is it, low density lipoproteins and things like that. So there are some lipids. And when they do come to the cell, they can get right through the plasma membrane. And then small molecules can diffuse across the membrane. Oxygen and carbon dioxide are the main two small molecules that move right across the membrane by diffusion. Generally, oxygen diffuses across the membrane into the cell, and carbon dioxide diffuses from inside the cell to outside the cell. Any question about any of that? All right, so the plasma membrane of prokaryotic cells have some special functions, like they have the enzymes for producing ATP. Now, in eukaryotic cell, this is not in the cell membrane. It's in the inner mitochondrial membrane. So it's in a membrane, but it's inside the mitochondria. And then in prokaryotic cells, we also have the photosynthetic pigments, which are responsible for photosynthesis. And that's where we find the chromatophores or thylakoids. Let me blow this up a little bit. So this is showing you an actual electron microscopic image of the plasma membrane of a photosynthetic cell. And that membrane folds into almost circles. It's not quite a circle because it'll be, what you call it, I don't know if I can draw it. I'll try and draw it with a mouse here, where the membrane comes across like that, and then it balloons out in a circle, and then goes like that. And I think I could do that. Did you guys understand that? Anyone not understand that? All right, I'm going to assume you're all understood it because I'm not hearing anything. But it then creates almost circles. They aren't totally circles. They're connected to the plasma membrane up here, and then it goes around, and then it's connected to the plasma membrane there. And these we call chromatophores or thylakoids. The thylakoid would be similar to what you find in the chloroplast, and that's why it's a slightly different name.
Do we need to know, do we need to identify those?
You don't need to identify those, but you should know that the plasma membrane of prokaryotes has the enzymes for ATP production. And then in the photosynthetic prokaryotes, this is where the pigments are, as well as the enzyme for at least beginning photosynthesis.
Thank you.
Okay. All right, damage to the plasma membrane will leak the contents of the cell, the cytoplasm, out. And you can damage the plasma membrane with alcohols, quaternary ammonium compounds, and then polymyxin antibiotics like polymyxin B. It puts a hole in the membrane, and then the cytoplasm can leak out. That will kill the cell. Any question about that? All right, the movement of material across the plasma membrane is critical to cells. There are two ways that movement can move across the plasma membrane. The first is passive, the second is active. So in passive processes, the movement of materials moves down its concentration gradient. That means things move from areas of high concentration to areas of low concentration. Like for example, if you were to eat a diet rich in sugar, diet rich in glucose, for example, outside the cell of your gut, would be lots of glucose. Inside the cell would be less glucose. And then the glucose could move across the plasma membrane in a passive process, moving down the concentration gradient. Now, that is not simple diffusion because the plasma membrane will not allow glucose to move through it. But you can have a protein that will facilitate the diffusion of the sugar. And I should change that from glucose to fructose, because fructose moves into the cell only by facilitated diffusion. And we'll talk a little bit more about that, meaning there's a protein that will facilitate that passive movement. And fructose only moves into the cell from regions of heavy concentration to regions of low concentration. And that would be like if you were to eat a diet high in fruit, you'd have lots of fructose in your gut, and it would move into the gut cell by facilitated diffusion. Okay. Another way is, of course, oxygen moves by simple diffusion, and it would move into the cell just by simple diffusion, right across the plasma membrane. Osmosis is the third way that material can move by passive movement across the membrane. And osmosis is unique in that instead of a solute moving, and simple diffusion and facilitated diffusion is when a solute moves across the plasma membrane. Osmosis is when the solvent moves across the plasma membrane. So osmosis is, what is that? In this case, we're talking about simple diffusion, but it is a solvent. And then osmosis is always moving across a membrane or a synthetic membrane. And it's always the solvent, like water in this case, moving into the cell. Of course, if the cell was in a dry environment, like you were to put a, I don't know, carrot in the desert, water would move out of the carrot into the desert sand, and that would be osmosis out of the cell. Any question about any of that? All of these processes are passive in that the movement of material is moving from areas of high concentration to areas of low concentration. The cell does not spend any energy to move the material. That's why it's called passive. Any question about that? All right. If not, let's move on to active. Active is the movement of material against the concentration gradient. For example, if you were to eat a diet very low in glucose, you could have a little bit of glucose outside your gut, and your gut cell would have more glucose in it. But the gut cell is going to say, well, I see a glucose molecule out there. I want to move that into the cell. And it will move that glucose from the gut in areas of low concentration to areas of high concentration. This is an active process, and it needs to require a transport protein to move the glucose into the cell against its concentration gradient. And it's going to need to spend energy to move the glucose or any molecule from areas of low concentration to areas of high concentration. Any question about any of that? A good example of this would be a amino acid. The cell is going to want to take an amino acid into the cell, even if it's at low concentration outside the cell and high concentration inside the cell, because we need amino acids to make proteins. And so the cell will use an active process to transport that amino acid into the cell against its concentration gradient. And that will cost energy to move each molecule into the cell. And the cell will do that by using up one ATP molecule, that's where it gets its energy from, to transport that protein using the transport protein, meaning the molecule has to move through a transport protein. Any question about any of that? All right, simple diffusion is just the movement of a concentration gradient down the concentration gradient. Like if you were to put a drop of dye in the beaker from areas of high concentration, it will decrease, it'll move to areas of low concentration. And that you can see happening. And eventually, if you would come back to this would be totally diffused. And so there wouldn't be a dark purple, and all the water would be light purple. This is important for the movement of oxygen and carbon dioxide across the cell membrane. Facilitated diffusion uses a transport protein, and this slide is calling it a transporter protein. It's the same thing as a transport protein, where it moves a molecule from areas of high concentration to areas of low concentration. And that's why it's called facilitated diffusion. The molecule is moving from areas of high concentration to low concentration. It doesn't require any energy on the part of the cell to move the molecule. And this is the way fructose moves into your cell, by facilitated diffusion, and by facilitated diffusion only. Meaning if you were to eat fructose, not all of it would ever be digested, because once the fructose equals, the concentration of fructose equals the concentration outside and inside equal each other. I didn't say that quite correct. Once the fructose inside the cell equals the concentration outside the cell, the net movement of fructose will not move. And so you'll never digest all the fructose. Okay? Because it only moves by facilitated diffusion using a transport protein. Facilitated diffusion can also happen with a channel protein, in which case there's a channel across the membrane. In both simple and facilitated diffusion, only the solutes are transported across the membrane, and that transport is always down the concentration gradient, from areas of high concentration to areas of low concentration. Osmosis is similar, but realize it's the net movement of water across a selectively permeable membrane. Water is not a solute, it is a solvent. And for osmosis to happen, it always has to go across the selectively permeable membrane, like the cell membrane or some man-made membrane, artificial membrane. Any question about that? So if I were to have a container of soil, and one part were to have dry soil and another part to have wet soil, and there would be no membrane in between them, that diffusion of water would be simple diffusion. It would not be osmosis, because there's no movement across the selectively permeable membrane. Any question about that? And so that would be the simple diffusion of water, which is kind of confusing, not osmosis. But then if I were to put a man-made membrane, just a, what do they call that? A polyethylene sheet that would allow water to cross, put it in between the dry soil and the wet soil, that would be osmosis, the net movement of water from areas of high concentration to areas of low concentration across the selectively permeable membrane. Remember that when we're talking about osmosis, the water is moving, but yet when we discuss the tonicity, we're not talking about the concentration of water. We're talking about the concentration of solute. When we're talking about the solute, wherever water is high in concentration, the solute concentration will be less. And wherever water is low in concentration, the solute concentration will be high. And that gives rise to three types of tonicity. A bacterial cell can experience three kinds of osmotic solutions. It can be an isotonic solution, a hypotonic solution, or a hypertonic solution. Realize these terms are relative terms, so it's correct to state the solution outside the cell was hypertonic relative to the solution inside the cell. And with a cell, it's pretty easy to realize we're talking about what's outside and inside the cell. And the tonicity is used in this case to discuss the solution outside the cell. The isotonic solution is the easiest to discuss. That's where the concentration of the solute outside the cell is equal to the concentration of the solute inside the cell. In an isotonic solution, there's no net movement of water into the cell. What I mean by net movement is for every water molecule moving into the cell, there would be one molecule leaving. So it's just as simple to say that there's no net movement of water into the cell. The water is moving, but realize the tonicity is what defines the solution. So we're saying not the concentration of water outside the cell is equal to the concentration inside the cell. It's the concentration of solute is equal outside the cell as what's inside the cell. The cell is happy in an isotonic solution. And if you were to put your red blood cells in an isotonic solution, the cell would continue to exist, okay? I used a red blood cell because it doesn't have a cell wall. In a hypotonic solution, the concentration of the solute is less outside the cell than the concentration of the solute inside the cell. So the water moves from outside the cell to inside the cell. And that will cause the plasma membrane to expand. And in a red blood cell, that plasma membrane would expand until it would burst. And then the cell would die because the contents of the cytoplasm would leak out of the cell. That is called osmotic lysis, when water moves into the cell, causing the cell to burst. Osmotic lysis can happen to a cell that doesn't have a cell wall, or a cell that has a damaged cell wall. Like if you give penicillin G to growing bacteria cell, it will cause a hole in the cell wall, and then the plasma membrane can protrude out of that hole and then would eventually burst. In a hypotonic solution, the water moves from outside the cell to inside the cell. Most bacteria live in a solution that is hypotonic. And that's why penicillin G is so effective at killing gram-positive cells. Any question about any of that? All right. A hypertonic solution is when the concentration of solute outside the cell is higher than the concentration of solute inside the cell. In this case, the water will move from inside the cell to outside the cell. This will cause shrinkage of the cytoplasm, and you can see the cell membrane pull away from the cell wall as water leaves the cell. This will cause plasmolysis, and that can kill the cell. If too much water leaves the cell, that will kill the cell. So for most bacteria, it will eventually lead to cellular death, like if an E. coli cell drops into a container of molasses, water will leave the cell, and then the cell will die by plasmolysis. And that is why molasses can stay at room temperature for years. I mentioned that my mother had a bottle of molasses that we figured was 20 years old, and I made molasses candy for my father, and he ate it, and he liked it, and he didn't die because there was no harmful bacteria in it, even though it was 20 years old. And that bottle of molasses lasted for 30 years because mother and I didn't really use molasses very much. And after about 30 years, my mother finally decided, you know, that molasses is old enough, I'm going to throw it away. But it looked perfectly good, okay, at 30 years old. Any question about any of that? And no, I don't recommend you keep your molasses for 30 years. Like I said, it was just because nobody used that molasses, and that's why it was so old. All right, enough about active transport. Passive transport. Now let's start talking about active transport. Passive processes are okay when the concentration of a substance required by a cell is higher outside the cell than inside the cell. Like for example, if you were to eat a diet high in fruit, then you'd have a high concentration of fructose outside the cell. But what about when a cell is in an environment with a low nutrient concentration? What's the cell going to do? Like a cell in the ocean, very little nutrients in ocean water, or a cell in the desert, very little nutrients in the desert. What the cell does is it uses an active transport to bring the nutrients into the cell. So the cell concentrates, accumulates nutrients against a concentration gradient. This requires energy for the cell to perform work. It costs the cell energy to move a molecule against its concentration gradient. The cell also uses an active transport protein to pump the solute molecule against its concentration gradient. So you require a transport protein, which is in the cell membrane. And generally, these transport proteins are molecule specific, meaning it will only move one molecule into the cell. And for those of you who know a little bit about drugs, we can have drugs that mimic the molecule the transport protein is supposed to transport. And so that molecule is not entirely the transport protein is not entirely molecule specific, but for our purposes, we can say that the molecule is one molecule specific, because we're not going to talk about drugs that interfere with transport proteins. I used to know a drug that interfered with the transport protein, and my mother used to take it, but I don't remember what it is anymore. All right, an active transport requires energy to move the molecule across the membrane using the transport protein. And how that energy comes about is the cell uses up one ATP. So it breaks down ATP and gets the energy from ATP. Remember, ATP is adenosine triphosphate. Any question about that? And for eukaryotic cells, this is the only way that they can move a molecule against the concentration gradient. You'll notice how I specifically said for eukaryote. For prokaryotes, they do have another way of moving molecules against their concentration gradient. And this is called group translocation. It's only found in prokaryotes. It does require a transport protein, so it is an active transport. It does require energy to move the molecule against its concentration gradient, but that energy usually comes from a high energy phosphate compound like PEP. We're not going to really talk about PEP. It could come from ATP, but it doesn't have to. And if you think about it, ATP is a high energy phosphate compound, okay? And this is one way that prokaryotic cells can transport glucose into the cell. They can transport glucose with group translocation. The difference between glucose and a prokaryote being transported by a transport protein and then those being transported by group translocation is that when the molecule is moved into the cell by group translocation, the molecule is chemically altered. For example, in the case of glucose moving into the cell, PEP transfers a phosphate group onto glucose. And so the glucose is changed. It's no longer glucose. It'll be glucose 6-phosphate. This alteration helps prevent the escape of the molecule from the cell, and it allows the cell to accumulate substances at a higher concentration than would otherwise be possible. Think about it. If there's 10 glucose molecules outside the cell and none inside the cell could use active transport and a transport protein to transport all of the 10 into the cell. But each time a glucose is moved into the cell, it'll be moving glucose against a higher and higher concentration gradient. And that would be more and more difficult. You see that? If, on the other hand, you were to transport 5 of the glucose into the cell with active transport using a transport protein, and you're to transport 5 of the glucoses into the cell using group translocation, you could move 5 into the cell, and that would mean 5 glucose are now in the cell. And then 5 glucoses would be moved into the cell by group translocation, and we'd have 5 glucose 6-phosphate in the cell. Now, if there were to be an 11th glucose outside the cell could more easily move that glucose into the cell because the concentration gradient inside the cell will not be as high if there is only 5 glucose as opposed to 10 glucose molecules inside the cell. And that's why group translocation helps cells accumulate substances. Any question about any of that? So remember, group translocation is very similar to active transport. The main difference is that it's only found in in prokaryotes, and it alters the molecule when it moves into the cell. Any question about that? All right, let's move on to the cytoplasm. The substance inside the cell is The cytoplasm, at least to prokaryotes, that's all you have inside the cell, unless it's something that doesn't dissolve in the cytoplasm. And we'll talk about that in a little bit. The cytoplasm is mostly water, 80% water with dissolved substances in it, and we call these substances solutes. This could be DNA dissolved in the cytoplasm. It could be glucose, it could be sodium chloride, it could be any of those solutes. But there are things dissolved in the cytoplasm. Now, in a eukaryote cell, the cytoplasm is all of the inside of the cell, with the exception of the nucleus. Inside the nucleus, we don't call it cytoplasm, we call it nucleoplasm. And we'll talk a little bit about that in tomorrow's lesson. The nucleoid region is wherever the DNA is floating around in the cytoplasm. And it can move because there's no boundary between the DNA and the cytoplasm in a prokaryotic cell. In a eukaryote cell, there is a boundary, and the boundary is the nucleus membrane. Inside the cytoplasm, we can find ribosomes as well, and that is a big protein that is so big that it actually is not in solution. So the artist has drawn it here, and you can sort of see the ribosome dots in the electron microscope image. And yes, there's lots of the dots because there's lots of ribosomes in the cell. There are also inclusions, which are accumulation of substances that so much of the substance has accumulated in one spot that that substance will literally precipitate out of solution. And then it forms what we call an inclusion. Something you can see under the light microscope, which I think that's an inclusion, and that's an inclusion. I don't have a link to it there, but there and there. The cell likes to accumulate certain stuff depending on what the cell does. And if too much of that gets accumulated in the cell, it will precipitate out of solution. That could be the cell is accumulating proteins. It could be other things like sulfate or iron. I've already talked about the nucleoid region, so let's move on. What am I talking about on this slide? Oh, ribosomes. I already talked about ribosomes, so let's move on. Let me mention that ribosomes are the site of protein synthesis. They are made up, ribosomes are made up of both protein, we call that ribosomal proteins, and then ribosomal RNA or rRNA. So a ribosome is both rRNA and ribosomal proteins. And there are subunits to the ribosomes. I'm not going to test you on that, so you don't need to know the subunits of the ribosome. You should know, however, that prokaryote ribosomes are smaller than eukaryote ribosomes. So we say that prokaryote ribosomes are 70S in size. The S is the unit that biochemists and molecular biologists use to measure ribosomes, and it stands for where the ribosome sediments in an ultracentrifuge gradient. So you spin this ribosomes in an ultracentrifuge, and you have a gradient, and the ribosomes will go to the 70th location in the ultracentrifuge. And the important thing to know, not that what 70S really is, but you should know that prokaryote ribosomes are smaller than eukaryote ribosomes. And we'll talk about eukaryote ribosomes tomorrow, but they're larger and they're 80S.
I have a question, Dr. Volpe.
Sedimented ADS, yes.
And what about tRNA?
We haven't talked about that yet. We're talking about ribosomes here. And so that's why I mentioned rRNA. When we talk about RNA, and I thought I already talked about RNA, there are three types. There's messenger RNA, tRNA, and rRNA.
Okay.
And this I'm just telling you is what the rRNA is for, making up the ribosome. Okay?
And then we don't have class tomorrow, right?
Tomorrow is Friday, so no,
Yeah.
you don't have class tomorrow.
Yeah, I think you mentioned a couple of times that you'll discuss the rest tomorrow. So
Oh, I meant in the next lesson, sorry.
no
Not
problem.
tomorrow. I meant in the next lesson. Yes, we don't have class tomorrow. You should correct me. Thank you for correcting me on that. Our next lesson isn't until next Tuesday, so. All right, inclusions are reserve deposits that are deposits in the cytoplasm, and so much of that material has accumulated that it actually precipitates out of the cytoplasm, meaning it's no longer soluble in the cytoplasm. You precipitate it out of solution. And if you've ever taken a bunch of salt in water, if you put too much salt or sugar in water, you put too much of the salt or the sugar in, eventually you put more and more in, it'll start precipitating out of solution. And that's what I mean. Too much of it's been in the cytoplasm, so now it starts precipitating out of solution. There are different types of inclusions, and that's all you need to know. You don't need to know the different types of inclusions, just know there are different types. And the type is just depending on what the molecule is that makes up the inclusions. Let's take a look at some inclusions. Magnetosomes, right there. And then this molecule metabolizes sulfur, so it's incorporating sulfur in the cell. And so we see sulfur inclusions called sulfur granules. And this is the metachromatic granules, which I think are proteins that have been built by the cell and form the metachromatic granules. Another thing you can see inside prokaryotic cells are endospores. Endospores are specialized resting cells produced by certain species when the environment becomes detrimental. These species tend to be in two families, family Bacillus and family Clostridium. These two families can make endospores. And an endospore is it's literally a non-metabolic cell, which is highly resistant to desiccation, heat, chemicals, radiation, almost anything you can throw at it. The endospore is resistant to it, almost anything. The endospore can survive conditions that the vegetative cells cannot. So here we have a parent cell and the environment has become detrimental. So this cell has made an endospore. You should realize that endospore formation is not reproduction. I think I've got that down here, so I shouldn't talk about it now, but I've already started. It's not reproduction because one parent cell makes one endospore. And then the parent cell dies because the environment has become detrimental to the cell. So 1-to-1 is not reproduction. Endospore is a survival trait. So the cell makes the endospore to survive. It's not a reproductive trait, it's a survival trait. Sporulation is the formation of the endospore. And then a germination is when the endospore germinates to return to a vegetative state. A cell without the endospore. And then the vegetative cell is a cell that's metabolically active and can reproduce and perform the normal things that cells do. So here we're seeing a picture of endospores, and they're being made inside a vegetative cell, the parent cell. And there's an endospore outside the cell. And what happened was its cell probably died, releasing the endospore. There's a vegetative cell, and it has not begun to form the endospore yet. Any question about any of that? All right, let's talk about endospore formation in just a little bit more detail, and then we'll watch that little video. I got it going now because, oops, go back to the slide. Let me blow this up. When an endospore starts to form, the cell goes through cell division in the normal process. So the DNA multiplies and then moves to one side of the cell and the other side of the cell. The difference is for bacteria, when an endospore forms, the cell doesn't divide in half, meaning the cell divides where one portion of the cell is larger than the other portion of the cell. The cell then begins to form the cell wall and the cell membrane in between the two DNA molecules. So the plasma membrane starts to surround the DNA and the cytoplasm inside the endospore, as well as the rest of the vegetative cell. And then the cell wall starts forming between the cell. The endospore gets two membranes around it. Peptidoglycan forms between the two membranes. So instead of being outside the membrane, it's actually inside the two membranes. And then a spore coat is put around the endospore. And there we can see the spore coat right there. And there's the endospore. And then when the environment becomes so detrimental, the parent cell that made the endospore will die. And that will release the endospore. Any question about any of that?
Yes, is that division happening by binary fission or mitosis?
No, it's by neither, meaning this is not binary fission, because binary fission would split the cell into half.
Yeah.
And this one is splitting it like something like 3/4 into 1/4. So the endospore is usually smaller than the other portion of the cell, which will be the continuation, I guess, of the vegetative cell, which will then go on to die.
Thank you.
Okay. And it's not mitosis, because prokaryotes don't have mitosis. It's literally called, I got it in there, sporulation, endospore formation. All right, let's watch the little video. Oh, shoot. Forgot it opens up the wrong page. So I got to copy that link. I think this is something new that Microsoft has done. So you can't open it directly from the PowerPoint. And a commercial is going to start off with.
Homeowner, 2022 is your last chance to go solar. In December of 2020, Congress passed an extension
All right, endospore formation. It's not a very good movie, but it does show you the process of endospore forming. And let's go back to that. Right there, if I can stop it. You'll notice that there's a little bit of the cell on this side and a little bit of the cell on that side. So the endospore is forming at the start of the cell, but as the endospore grows, it takes up all of this side of that cell. All right, any question about endospore formation? If not, let's just begin talking about eukaryotic cells. You need to know the cell structures of eukaryotic cells. And if I don't finish this, it's okay, this lesson, because you have had this lesson in your previous class, but it's showing you the structures in a plant cell and in an animal cell in this slide. Then you'll notice that there's many things that are the same, like the nucleus is in both plant and animal cells. Actually, the flagellum can be both in animal cells and plant cells. It is true it's more likely to be seen in an animal cell, but you can have a flagellum in plant cells. And the Golgi complex is found in both plant cells and animal cells. There are a few things that are only found in one or the other, like chloroplasts are only found in photosynthetic cells of plant cells. So you will not find a chloroplast in the root cell, or the roots, or a root cell, and you won't find it in an animal cell, but you will find it in the leaf of a plant. Any question about that? Another thing that you can find in a plant cell, and it's not labeled here that way, but it's a central vacuole, is only found in a plant cell. Now, vacuoles can be found in a plant cell and in an animal cell. The vacuole isn't on here, but you can find a vacuole in an animal cell. And then there's something found in an animal cell, but not in a plant cell. And let me see if I can remember what it is. It's the, there it is. This structure right here, the centriole and the paracentriolar material, it makes up in an animal cell the mitotic spindle, if you remember from biology 160. So you can find this structure, I blow that up, right there in an animal cell, but it's not found in the plant cell. And it's curious because a plant cell does have a mitotic spindle when the cell goes through mitosis, but the plant cell never has a centriole. All right, any question about any of that?
So the vacuole is only found in the plant cell, not in the animal cell. Is that correct?
No, the central vacuole is only found in plant cells.
Okay.
Vacuoles are found in a plant cell and in an animal cell. This picture is showing you the central vacuole, not a vacuole, because I can tell because it's so large. A vacuole is this size or smaller. Okay.
Thank you.
All right. And we'll talk about that later in our next lesson. We won't have time to talk about the differences between central vacuole and a vacuole. For a plant cell, the central vacuole and the vacuole do the same thing, but let's see. I think that's about it. Eukaryotic cells are larger than prokaryotic cells. Remember, they're about 10 times larger. And here we're looking at an actual plant cell on this side in the electron microscopic image, and an actual animal cell on this side, also in the electron microscopic image. Oh, well, there's a vacuole. But right there, see that big thing there? That's the central vacuole. And there's no central vacuole in an animal cell. Shrink that down. So this is showing you a little bit of the differences between a prokaryotic cell and a eukaryotic cell. The cell size is different, about 10 times smaller for a prokaryotic cell. There's no nucleus in a prokaryotic cell. There is in a eukaryotic cell. There's no membrane-bound organelles in a prokaryotic cell, and there are membrane-bound organelles in a eukaryotic cell. The flagellum differ between the two, but let's not really talk about the differences. Both can have flagellum. The glycocalyx is present in both the prokaryotic cell and the eukaryotic cell. The cell wall of a prokaryotic cell is usually chemically complex. Its main molecule is peptidoglycan, but it has other molecules in it. And the eukaryotic cell wall is chemically simple. Like plant cells only have cellulose in the cell wall. The plasma membrane between a bacteria and a eukaryotic cell is about the same. The exception is an animal cell has sterols in it and carbohydrates, and these can serve as receptors. Sterol also serves as strengthening this plasma membrane. Prokaryotes don't have carbohydrates in their plasma membrane. You know, that's not entirely correct. I'm going to look that up. Of course, it's on the cell wall, so maybe it isn't in the plasma membrane. And then it generally lacks sterols. The cytoplasm, this is a little bit out of date, the book. It's been known for at least 10 years now that prokaryotes do have a primitive cytoskeleton, but it's primitive compared to the extensive cytoskeleton of a eukaryotic cell. And then eukaryotic cells have cytoplasmic streaming, which is absent in the prokaryotic cell. The ribosomes are present in both, but the ribosomes are smaller in prokaryotes, they're 70x, and they're larger in a eukaryote. We'll talk about the organelles having smaller ones later. The chromosomes are usually different. The DNA does have proteins in both the prokaryote and the eukaryotic chromosome, but the prokaryotic chromosome lacks the protein histone. And we're talking about bacteria here, not archaea. So we're talking about bacteria. So this isn't quite correct. It's not prokaryote here, it's bacteria. The chromosome lacks the histone protein. And you can find multiple histones on the chromosome, many histones on the chromosome of a eukaryotic cell. And generally, there's multiple linear chromosomes in a eukaryotic cell. And in a prokaryote, it's usually a single circular chromosome. The cell divides differently. The prokaryotic cell divides by binary fission, and the eukaryotic cell divides by mitosis. Sexual recombination, generally none in a prokaryotic cell. They can transfer plasmids, so DNA in a plasmid. And then sexual recombination happens in meiosis of eukaryotic cells. All right, any question about any of that? So prokaryotic cells can move in more ways, eukaryotic cells can move in more ways than a prokaryotic cell. I think I started saying that incorrectly. Eukaryotes can move by a flagella, which is the only way that a prokaryotic cell can swim. But eukaryotes can move by flagella, by cilia. The difference is if there's many of them on the cell and they're shorter, this is a cilia. If there's a few and they're long, a whip-like structure, that's a flagella. Eukaryotic cells have a third way of moving, and it's by putting out pseudopods. And so this cell, what's a good example? Let me shrink this down. There we go. Nope, wrong way. Gotta shrink it down even further. There we go. This cell here, so I'm removing the flagellum, can move this way by putting out its cytoplasm on this side and then pulling up cytoplasm on that side. And that's how the cell can move by pseudopods. And we'll talk a little bit about that later. Only cilia and motion by pseudopods happen only in eukaryotic cells. The cell wall can be present in eukaryotes. You generally find the cell wall in plants, algae, fungi. All of these cells also have the plasma membrane. However, the cell wall, if it's present, is chemically simple. Like for plants and algae, it's the only one we're going to talk about. The cell wall is only composed of cellulose. Okay, we're not going to talk about the cell walls of fungi and yeast. They're made up of something other than cellulose. Remember that there's no cell wall in animal cells. Instead, the plasma membrane may be covered by a glycocalyx. And then there'll be carbohydrates and proteins inside the lipids of the plasma membrane of animal cell. The cell membrane of eukaryotes is similar to bacteria. There are some differences. The main molecule of the cell membrane is the phospholipid, and the phospholipid does get together to form a bilayer, and it does contain peripheral and integral protein, including transmembrane proteins. The cell membrane does show selective permeability, and it can have transport across the membrane. And in this sense, it's the same. It does have simple diffusion, facilitated diffusion, and osmosis, but it only has active transport. There is no group translocation across the cell membrane of a eukaryote. There's one other way that eukaryotes can bring material inside the cell or outside the cell. Unlike prokaryotes, eukaryotes can move material across the cell membrane using vesicle transport, where a vesicle will form and then fuse with the cell membrane and dump its contents outside the cell. or contents outside the cell can form a vesicle inside the cell, and then the cell can bring the contents that were once outside the cell inside the cell. Any question about any of that? There are two types of vesicular transport. Endocytosis, that's the movement of material inside the cell. And it will happen by this vesicle. There are two types, phagocytosis, literally where the cell will eat. Pseudopods will come out of the cell. See if I can draw that. Got too many windows open. Yeah, it's going to be a little small, but it's all right. So here we have a cell. And we're going to put a little material right here. The cell can put pseudopods out, and these will grow around that material. Until the pseudopods totally surround the material. Come on, I'm not able to draw that. And you got to connect those together. I'm not going to do that because the eraser will be too big. And then this will be brought into the cell by a vesicle, which is membrane-bound organelle. And there will be material, which I'm going to just draw it as a line, which used to be that stuff outside the cell. Any question about that? The other way that a vesicle can form is where the material will be on the membrane, and then the membrane will literally start folding in on itself. So here's the cell. Something can be on the membrane. The membrane will then start invaginating, and that material will be brought into the cell, and then the invagination will connect. So the cell membrane will reform, and it will form a vesicle inside the cell, and then the vesicle will move inside the cell. This can be called cellular drinking, and it's termed pinocytosis. Any question about phagocytosis and pinocytosis? Both of those are endocytosis. That's different from exocytosis, where material moves out of the cell via a secretory vesicle. Let me get the window again. Here we have a cell. And we can make our material inside a vesicle. This vesicle can then move up to the cell membrane. And then the membrane of the vesicle can fuse with the membrane of the cell membrane. And then the two will be together. We're just going to draw it like this right here. Come on. And then that vesicle can open up. And let's erase this right here. And the lower portion connects to the vesicle. So there it is, the cell membrane. And then that'll dump the contents inside the vesicle, which can then go out of the cell. And that's exocytosis. Any question about any of that? All right. If there's no questions, I'm going to end the presentation here, and I will see you in the lab at 6:30. Okay, I'll see you later. Didn't seem to get that.